Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry faculty. In this video, we are going to discuss about biological oxidation. So let's start up with the discussion on biological oxidation. Now see, as it is clear from the word itself, biological oxidation means oxidation of the biomolecules in the body. Because always remember, we have basically three things which we eat in our diet. We eat carbs, we eat fats, we eat proteins. These are the things which we have in our diet, along with obviously vitamins and minerals which are used as the supplements. So these carbohydrates, they are converted into acetyl coenzyme A. Fats are converted into acetyl coenzyme A, and ketogenic amino acids are converted into acetyl coenzyme A. Now the question is that whatever we are eating in our diet, be it the carbs, be it the fats, be it the proteins, they all are being converted into this acetyl coenzyme A, which is having 2 carbon atoms, and this undergoes TCA cycle. Right? Some processes are taking place in the cytoplasm, some are taking place in the mitochondria. Let's say if I talk about carbohydrates form pyruvic acid, this is taking place in the cytosol. If we talk about how fats are converted into this, it is taking place in the mitochondria. So depending upon this, and TCA cycle always takes place in the mitochondria. Now the question is, what do you mean by the term biological oxidation? Thing is that biochemistry is a typical, I would say all the reactions are typical redox reactions. And what are redox reactions? They are the ones where oxidation as well as reduction takes place simultaneously. So if something is being oxidized, means if somewhere there is a loss of electrons, on the other side there will be something to gain those electrons. So leading to a redox reaction. So what happens here is, as you have noticed in fact all the videos wherever we have discussed about the metabolism, you must have noticed this terminology. NAD is converted into NADH plus H positive, and FAD is being converted into FADH2. Right. What do you mean by this positive charge? Means H positive and H2. So basically what is happening inside the body is, whenever we are eating something, the body undergoes a well-fed state, and whatever is there, it is consumed and stored. But whenever the body is performing something, that something is broken down to release energy. That is what is the first law of thermodynamics also, that energy can neither be created nor destroyed. It is simply converted from one form to another. Okay, so what is the use of these reducing equivalents? We know glucose is forming pyruvic acid is forming acetyl coenzyme A. When one molecule of glucose is oxidized, it is giving some 32 molecules of ATP. Similarly, one molecule of palmitic acid on oxidation is giving so many molecules of ATP. So what happens to these reducing equivalents? Nothing is wasted as such. These, since they are positively charged, so they are going to combine with the electrons in the presence of oxygen, because it is biological oxidation. Oxygen is involved, forming water. And this water, so produced as a result of metabolism, is known as metabolic water. And whatever energy is released during the transport of these protons, that energy is required to produce ATP, or it will simply lead to the production of ATP, and we know ATP is the energy currency of the body. Just say whenever if we go to the market, we spend something to buy. Same is the case with the body. Body will give something in the form of ATP and body will get energy. So biological oxidation mein there are two processes which are taking place. One is the electron transport chain. As it is clear from the word, there is transport of electrons. plus synthesis of ATP. And synthesis of ATP means phosphorylation. This process is known as oxidative phosphorylation. So, these two processes are coupled to form biological, they will be, the combined terminology will be biological oxidation. So, I can say that biological oxidation is electron transport chain plus synthesis of ATP, which is known as oxidative phosphorylation. So, in this video of ours, we are going to discuss what is this electron transport chain, how this chain goes on, and how the formation of ATP takes place. Where this entire process is occurring, what is the significance of this, right? So, let's start up with first of all electron transport chain. Now, see what happens, that this electron transport chain is basically the transport of the electrons along with the H positive ions. Now, where it occurs, so the site is the inner mitochondrial membrane. So, I can say that electron transport chain takes place in the inner mitochondrial membrane. And it consists of 4 main complexes. 4 main complexes. Now, if we talk about the first complex, complex 1 is NADH coenzyme Q reductase. It is going to bring electrons from NAD, or you can say NADH plus H positive, like that. And there are so many enzymes which are undergoing this NADH coenzyme Q reductase. Okay. Then comes complex 2. And this complex basically transports in the form of succinate coenzyme Q complex or succinate coenzyme Q reductase. And if we talk about the enzyme here, the enzyme here is succinate is converted to fumarate and FAD is converted into FADH2. And if you remember, when we discussed TCA cycle, I told you one very important thing, that TCA cycle mein all the enzymes are present in the matrix of mitochondria except SDH, because SDH is the only enzyme which is present on the inner mitochondrial membrane, right? Then complex 1, complex 2, and both of them give their electrons to a common acceptor. And what is that common acceptor? Common acceptor is coenzyme Q. Coenzyme Q, which is also known as ubiquinone. Why ubiquinone? Because it is ubiquitous in nature. And then comes complex 4. Complex 3, right? Complex 3 consists of cytochrome B(C)(1), cytochrome B to cytochrome C1. And what are these cytochromes? These cytochromes are the iron sulfur proteins. Right, very, very important. And this is the basic difference between cytochromes and heme. Yeah, iron present in heme. This iron here exists in 2 forms, ferric and ferrous. Okay, And we know that hemoglobin mein heme, which has iron, it has iron only in the ferrous form, never in the ferric form. But cytochromes are iron sulfur proteins which are having iron in both the forms, because obviously if something is being oxidized, if something is losing electrons, the other species should be there to gain those electrons. And then comes the last complex, complex 4. And this complex is a very, very important complex which is actually accepting the electrons, cytochrome A1, A3 oxidase, where actually the formation of water takes place. This is the site where water is synthesized, right? So these are the 4 major complexes of electron transport chain where there is transport of electrons. Now the question is how these electrons are moved, because there are high energy compounds in the body. Okay, and there are different type of shuttle systems, because ultimately it has to occur on the inner mitochondrial membrane. But the only thing is that there are 2 shuttles which are providing these equivalents to the mitochondria, because you have seen there are processes which are occurring in the cytoplasm. For example, if I talk about glycerophosphate shuttle, glycerophosphate shuttle mein it is occurring in the cytoplasm, so it has to enter into the mitochondria. Malate aspartate shuttle, again it has to come to the mitochondria. So wherever these reducing equivalents are produced, they will have to enter into the inner mitochondrial membrane, where they will undergo these 4 complexes. And the most important point to remember here is that they move as per their redox potential. And what is this redox potential? It is simply the potential or the ability to lose or gain electrons. And the most important point which we have to remember is complex 2 has very low redox potential. Very important. And just because of this reason, we always say that one molecule of FAD will always give only 1.5 ATP molecules on hydrolysis, whereas one molecule of NAD will give 2.5 ATP molecules. Right? So this is redox potential. Now we are going to study how these different complexes are acting, where actually this is present. So say this is complex 1, right? This is complex 2, right? They will hand over their electrons to coenzyme Q. This, and this coenzyme Q will give its electrons to complex 3 and finally in the end complex 4. Okay, so here complex 3 is cytochrome B to cytochrome C1, cytochrome C, and this is cytochrome A1, A3. And here I can say water is produced. And obviously this reaction is occurring like this. Okay, now if we talk about, and if I'll just show you a pictorial representation of this. Say this is the outer mitochondrial membrane. Right, and this is the intermembranal space like this, and this is the inner mitochondrial membrane. So what is happening here? Here this is complex 1, here complex 2, complex 3 and complex 4 like this. Okay. So this is one which is handing over to coenzyme Q like this. This is complex 2. This is 3. This is 4. Cytochrome C. And here we can say water is produced. Now the thing is, it will be releasing protons here. So here 4 protons and 2 protons will come. Right. Now the question is that there will be exchange, or you can say there will be simple a flux of protons which will be generated here. Now the question is, a kind of concentration gradient will be obtained here. This has to move. Up problem with them is these protons are impermeable to this mitochondrial membrane. They cannot cross this mitochondrial membrane. So they require an exit, because I told you that electron transport chain is occurring where due to the transport of electrons, energy is released at every step. So this energy, either it can be lost in the form of heat or it can be utilized to synthesize ATP. There can be only two options. Now the thing is that it is coupled with the synthesis of ATP. Otherwise, if all the amount of energy is liberated in the form of heat, there won't be any synthesis of ATP. Okay, so oxidative phosphorylation will not be achieved. Right? So what happens is, due to the release of these protons, यहाँ पे there is a kind of concentration gradient which is there. Now this has to move. Since these protons are impermeable to the mitochondrial membrane, they require, they get a transit with the help of F1, F0 particles which are present on the mitochondrial membrane like this. And if I show you the structure like this, say if this is the mitochondria, we know that inner mitochondrial is invaginated to form crystal type of structure. Right? Here, these F1, F0 particles are there. And these are known as F1, F0 particles are known as oxysomes. And they have a head, they have a neck and a base. Okay, so these protons will find an exit to move here, and here synthesis of ATP will take place. And these are the oxysomes, or they are known as the lollipops of mitochondria. Right? I'll just repeat it again. What I am trying to tell you here is there are two processes which are taking place. What is happening is electron transport chain mein there is transport of electrons at every step. Now when the electrons are being transported at every step, they are going to release the energy. And how that energy is maintained? Because that energy is maintained by the increased concentration of these protons. These protons are impermeable to the membrane. They cannot enter the membrane like this. So they require, this is actually known as Chemiosmotic hypothesis, which was given by Mitchell scientist also. There were other hypotheses also, but the most widely accepted was given by Mitchell. So they will find an exit through this, where they are going to form this ATP in the presence of ATP synthase complex. Right? So if I have to explain it, I will simply explain it in the form of simple steps, that release of protons will lead to an increase in the concentration of H ions. right? So they have to move along the concentration gradient. They have to move along the concentration gradient, which means high to low, right? So at every step there will be, or you can say they are impermeable to mitochondrial membrane. This is also a very important point. They are impermeable to the inner mitochondrial membrane. So a kind of force is generated there. So a kind of PMF is generated, proton motive force, or PMF is generated. And this theory, chemiosmotic theory, given by Mitchell. Right? So the moment they will move, they are going to exert a kind of energy which will be taken over by these F1, F0 particles. Right? So a kind of proton motive force is generated and they make an exit by F1, F0 particles, right? And ATP synthase is already, actually there are different type of subunits there, but again you don't have to go into that much detail. You just have to focus on one thing, that this ATP synthase activity is there, the enzyme is there. So I can say what is happening is, these are the oxysomes, right? They will come, which will be gained by ADP plus inorganic phosphate. This is the energy and it is going to form ATP. And the name of the enzyme is, this is the base, this is the neck, and this is the main one. So yahan pe you can say F1, F0 particles are there. In the presence of ATP synthase, the synthesis of ATP occurs. Okay. So this is very important to understand. First point is, release of protons will lead to an increase in the concentration of protons. They have to move along the concentration gradient, obviously, from high to low. But the only tragedy with them is they are impermeable to the mitochondrial membrane. So they have a kind of alternative, and that alternative is the F1, F0 particles present along the inner mitochondrial membrane, and they just make an entry into that. And this was chemiosmotic hypothesis also. Chemiosmotic hypothesis means there is increased concentration of the protons in between. You can see the space, and in the diagram it is clearly showing that these protons are generated there. So they will make an entry like this, and that is why we always say that if we talk about this is complex 2. Complex 2 is having low redox potential. So that is why it will give only, means one molecule of FAD will only give 1.5 ATP, whereas one molecule of NAD will give 2.5 ATP on hydrolysis, and that is why we always write NADH plus H positive and we write FADH2. Okay, so this is the synthesis of ATP. Then the process of synthesis of ATP is known as process of synthesis of ATP is known as oxidative phosphorylation. Oxidative phosphorylation. phosphorylation, which is different from substrate level phosphorylation, right? Different from, I can write it also, which is different from substrate level phosphorylation. Right? Now, next is, this is how electron transport chain leads to the synthesis of ATP. Then after this, then we come over to the again another important segment, what are the inhibitors and what are the uncouplers of this electron transport chain and oxidative phosphorylation. Because the most important point which you have to remember here is both the processes are coupled. What do you mean by coupled? That whatever energy is being lost, that will be required for the synthesis of ATP. So I can say that this 𝗖𝗵 is coupled with oxidative phosphorylation. So there are two terminologies here. One is the inhibitors. So inhibitors can be of 𝗖𝗵. Inhibitors can be of oxidative phosphorylation. But there is one more term which can uncouple these processes, and that will be known, they will be known as uncouplers. Okay, so there is a difference between the inhibitor and the uncoupler. Inhibitors are going to inhibit a particular process or the step or the complex, whereas uncouplers are going to uncouple the two processes, right? So first of all, we are going to take up the inhibitors of electron transport chain. Inhibitors of 𝗖𝗵. Now just now I told you 𝗖𝗵 has 4 complexes. So we can say that complex 1 is there, which is NADH coenzyme Q reductase complex. Complex 2 is there, which is succinate FADH₂ or simple you can say succinate coenzyme Q. Main acceptor coenzyme Q or ubiquinone. Complex 3. And finally complex 4. Now if we talk about the inhibitors here, the inhibitors of complex 1, complex 2, complex 3 and complex 4. Complex 1 के inhibitors rotenone, which is a common fish poison, right? Second is 𝗵𝗵. And third is, you can say, one is rotenone 𝗵𝗵𝗵𝗵. 𝗵𝗵𝗵𝗵. Okay. Complex 2 carboxyl, malonate and TTFA, trienoyl trifluoroacetone. Trienoyl trifluoroacetone. If we talk about the complex 3 inhibitors, the complex 3 has generally all the hypoglycemic drugs like metformin is there. Second is BAL, which is British anti-leucide. British anti-leucide. And third is antimycin. Then we come over to complex 4. Now complex 4, the main inhibitors of complex 4 are carbon monoxide, The most important one, cyanide, sodium azide, and H2S, hydrogen sulfide. Okay, so we can say that these are the inhibitors of electron transport chain. So complex 1 has rotenone, pyridine, 𝗵𝗵𝗴𝗍. Complex 2 has carboxyl, malonate and TTFA. Complex 3, metformin, BAL and antimycin. And complex 4 mein the most important one is cyanide, carbon monoxide, sodium azide and hydrogen sulfide. So these are what? These are the inhibitors of ETC, right? Then we come over to main inhibitor of oxidative phosphorylation. Phosphorylation. Now here the most important inhibitor is mainly the antibiotics. Let's say if I talk about oligomycin. Oligomycin is an inhibitor of ATP synthase. Right? There is one more inhibitor, we call it as 𝗃𝗫𝗫. This 𝗃𝗫𝗫 is an inhibitor of translocase. So I can say ATP translocase. Right? Then another terminology which I used was uncouplers. So what are the uncouplers? These were the inhibitors of the two processes. सबसे पहले तो how do we define these uncouplers? So uncouplers are the molecules which can uncouple the two processes. And what are these two processes? ETC plus oxidative phosphorylation. And how they will uncouple? They will release the energy in the form of heat. So I can say that no ATP will be formed when uncouplers are there. So no synthesis of ATP will take place. ATP will take place. Very, very important. Right? So these are the uncouplers which can uncouple the two processes. So whatever energy is released which was used for the synthesis of ATP, that will be dissipated in the form of heat. So I can say that ETC plus OP. So uncoupler will uncouple and the energy is dissipated as heat. And these uncouplers are of two types. Uncouplers are of two types. One is the synthetic uncouplers and the other one is natural uncouplers. Now what is the difference between synthetic and natural uncouplers? Synthetic will include dinitrophenol, 2,4-DNP, dinitrocresol, and aspirin if taken in high doses. Aspirin in high dose. And if we talk about the natural uncouplers, the most important natural uncoupler is thermogenin. the free fatty acids. Right? Thyroxine. They are the natural uncouplers which are already present in the body, which are actually regulating the temperature of the body. Okay, but this 2,4-DNP, it was used as a, basically it was banned by FDA. Why it was banned by FDA? Because it was used as a weight reduction drug or weight reducing drug. Right, and if we talk about the natural uncouplers, thermogenin and all are very, very important because, you know, when the child is born, child has very chubby cheeks. Right, similarly, so they have more brown fat. Usme there is no use of synthesis of ATP. So electron transport chain will be there. So it will take place at a normal place, but the most important thing is ki wahan pe the energy will not be utilized for the synthesis of ATP. No oxidative phosphorylation will take place. Similarly, if we talk about the hibernating animals, जैसे rabbit and all, rabbit has such furry type of body. So what happens is so much furry body is there, so it has more of brown fat. So उसमें क्या है? Oxidation is more, oxidative phosphorylation will not take place. So it's like you can say that कि thermogenin and all, या the natural couplers, can make the body warm, can make the body warm as the heat is dissipated. Or you can say no synthesis of ATP is there. No synthesis of ATP takes place. And we can quote the example of hibernating animals. Example can be hibernating animals and the newborn babies. Right? So this is what is the entire metabolism which is linked by electron transport chain. The energy is required for the synthesis of ATP. So whatever we are eating, it is coupled. Coupled to what? ETC plus oxidative phosphorylation. Right? That is why electron transport chain and OP are the very, very important, I would say, topic from examination point of view also, from clinical point of view also, because if we talk about the inhibitors, as I told you, cyanide poisoning leads to what? The individual dies. Why he dies? Because the complex 4, the ultimate complex, which is actually accepting all the electrons and protons to form water, it is blocked. So it leads to the death of the individual. Okay, and in some books it is written as this ATP synthase is complex 5. So in some books you will find it as that the entire, you can say, biological oxidation mein electron transport chain consists of 5 main complexes. 1, 2, 3, 4 and 5th one is quoted as ATP synthase complex. And you have to understand what is the difference between uncouplers and inhibitors. Inhibitors are going to block a particular complex, whereas uncouplers are going to uncouple the two processes. So whatever energy is utilized for during the transport of electrons, that will be dissipated in the form of heat if uncouplers are used. Right? So this is all about the video on biological oxidation. I hope this video is clear to all of you. Thank you so much.